In cell culture, maintaining and passaging adherent cells requires enzymatic detachment. Trypsinization is a widely used method, but beyond its routine use, understanding its underlying mechanisms is crucial for optimizing experimental outcomes. In this video, we will break down the biochemical principles behind trypsinization, discussing why each step is necessary for ensuring cell viability and reproducibility. Adherent cells attach to culture surfaces via integrins, which link them to extracellular matrix proteins. These interactions are stabilized by divalent cations such as calcium and magnesium. While adhesion is essential for proliferation and function, it poses a challenge when cells need to be passaged or reseeded. Trypsinization facilitates detachment by enzymatically cleaving cell adhesion proteins, allowing cells to be transferred and expanded. Trypsin is a serine protease that hydrolyzes peptide bonds in proteins, specifically targeting lysine and arginine residues. By digesting focal adhesion proteins, it disrupts cell attachment to the culture surface. However, the presence of calcium and magnesium stabilizes these adhesion complexes. To enhance trypsin efficiency, EDTA is included in the solution to chelate divalent cations, further destabilizing cell adhesion and making enzymatic cleavage more effective. Before introducing trypsin, cells are typically rinsed with PBS. This step serves two main purposes. First, it removes residual serum, which contains trypsin inhibitors that could prematurely neutralize enzymatic activity. Second, it clears metabolic waste and dead cells, ensuring a more controlled detachment process. Once trypsin is added, incubation at 37 degrees Celsius accelerates proteolytic activity. The duration is critical. Insufficient exposure results in incomplete detachment, while prolonged digestion can damage cell surface proteins essential for reattachment and viability. Regular microscopic observation helps determine the optimal endpoint, where cells appear rounded and start to lift from the surface. As trypsin degrades adhesion proteins, cells lose their anchorage and undergo cytoskeletal reorganization, transitioning from a spread morphology to a rounded shape. This change is a hallmark of successful detachment and can be monitored under a phase contrast microscope. To prevent overdigestion, trypsin activity must be promptly inhibited. This can be achieved by adding a complete culture medium containing FBS, which provides protease inhibitors, or by using a dedicated trypsin inhibitor. This step is crucial for preserving cell membrane integrity and ensuring optimal post-passaging recovery. Post-detachment, cell suspensions often contain aggregates that need to be dispersed for accurate seeding. Gentle pipetting facilitates uniform distribution without inducing excessive shear stress, which can compromise cell viability. The goal is to achieve a single-cell suspension while maintaining physiological conditions that support survival and proliferation. Trypsinization is more than just a routine procedure. It is a precisely regulated process that balances enzymatic activity, ion chelation, and mechanical handling to maintain cell health. A deeper understanding of these principles allows for better protocol optimization and improved reproducibility in cell culture experiments. Thank you for watching. If you found this discussion insightful, stay tuned for more advanced cell culture techniques and optimization strategies.